Hi, this is Miss Litton, and this is my wonderful Period 1 AP Bio class. Say hi. Hi. They are excited. Okay, so we're going to finish up in Chapter 12, and we are talking about how, at this section, how code in our DNA will translate into a protein. So this is all underneath our molecular biology of the gene, and I believe we are in section 12.3, the genetic code of life. Yes? Okay, and you have all the notes, so this time I'm not going to go back through and give you the notes since you have them, right? Okay, so hang on. And I think we did this slide, yes? A little bit? No? Okay, well, I'll walk you. Okay, good. All right, so here we go. So you have a sequence of nucleotides on your DNA. Those nucleotides are going to code for a series of amino acids. What do amino acids build? Proteins. So could that protein be a specific enzyme? Yes. Okay, and could that enzyme then determine the functionality of that cell and that organism? Yes. So the DNA is coding for the amino acids hook together in order to form proteins. If you put the amino acids in the wrong order, you will have the wrong what? Protein generated from that, okay? That sequence is very important. So a little reminder about the differences between DNA and RNA. Blue, could you handle that real quick? Yes. All right, so RNA is single-stranded nucleotides, and the sugar is different. The sugar is what? Ribose, whereas DNA it is in deoxyribose. And all the bases are the same, except wherever you would think, oh, a thymine will be there, instead a uracil is there. Good, and it is single-stranded, not double-stranded. Here, in essence, is how you build a protein. So make sure you're watching me. Your eyes are up, okay? Because I'm going to power right through this. Up at the very top, you can see DNA. DNA is a double what? Helix. Before, when we were making more DNA, we had to do what? We had to do the topoisomerase. We had to do unwind and unzip. And what did we have? Helicase. And then we had to do what to hold them apart? SSB. Good. We had a strand and a strand. What are they? Leading and lagging. RNA is single-stranded. So do we need both of these? No. What strand could we put away? Put the lagging strand away. And now, instead of using DNA polymerase to boom, 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 what could we use instead? RNA polymerase to boom, boom, boom. And when we use our RNA polymerase, if there's a G on the DNA, what are we going to put here? G with C, C with G, A with U. We're making RNA, okay? Think it out. A with, we'll bring in a nucleotide that has uracil in it instead, right? And then T with A. You still have adenine. Just wherever you would want to put a thymine type nucleotide, you're going to need to put a uracil. Okay? Are the phosphate sugar backbones going to be different? Yes, because before it went deoxyribose phosphate, deoxyribose phosphate, deoxyribose phosphate. This one is going to go what? Ribose phosphate, right? Okay, so the backbone will be different because the sugar is different. Every nucleotide, when we boom boom, okay, we'll have ribose instead of deoxyribose, and wherever we'd want to put a thymine instead, we will put a uracil. We will then form, okay, we will form some RNA. You can see here, it's only using one strand of the DNA, and that one strand is the leading strand. Now, keep in mind, if I'm holding up this book, and this entire book is my genetic code, and I need a protein, okay, I don't need to copy the whole book. If I was making another AP Bio book, another whole copies, that would be DNA replication, right? And that's what we learned before. Now, what I need is I need the information on how to make insulin. It's located right here. I'm now going to make an RNA copy of how to make the DNA can zip back up and stay in the nucleus. I now have the information, and this is, what is this called? mRNA, right? And I can take this mRNA, and what does the M stand for? Message. Yeah, messenger. And this is what I'm going to use to build my protein. Now, you can see here it says the words mature RNA, and let me explain why. Our DNA has a lot of genetic junk in it, failed attempts at evolution. Okay? It has structural components just on the end of the chromosome that help in the structure of it that doesn't code. There are non-coding sections. And so when you make your message, you need to clean out all the junk that we don't need, or the failed attempts, or just the structural. That's why it says mature RNA. You literally use spliceosomes, little snurps, and you're going to remove the parts we don't need. And those parts are actually called, annoyingly, introns, right? Super annoying because it's in. The parts you leave behind are called exons. Again, super annoying. But the reason why they're called exons, the part that remain, is because that's the DNA that gets expressed. Okay, expressed. And so you cut out these little sections you don't need, and then you protect either end. Because this is a naked little single-stranded pieces of RNA that's going to move out into your cytoplasm if you're a eukaryotic cell. There are enzymes out there that could digest it, so you protect it. On one end, you put a cap of 7-methylguanosine. That's a little protection. And on the other end, you put a poly-A tail. It's about 100 to 200 adenines. So if some enzyme comes along, it's just biting off little ends of the tail, and it's not interfering with any of the coding sequences. That's what a mature RNA is. A cap, a tail, and you cut out the introns and leave the exons. So when we see mature RNA, that's what's going out here into the cytoplasm. Now, where does this mature RNA need to go? It needs to go to the workbench of protein synthesis. Where is the workbench? You know this. What's the workbench of protein synthesis? A ribosome. So it's going to go out here, and it's going to sit its happy body right here on a ribosome. Now, every 3 bases on this mRNA right here, every 3 bases is called a codon. Every 3 bases is a codon. If this codon says CCC, what would be complementary to CCC? GGG. So, tRNA, what does the T stand for? Transfer. How do you know which tRNA needs to land there? The one, if the codon says CCC, then the transfer of the tRNA has to be what? GGG. And what is it transferring? It's transferring an amino acid. So right here is a tRNA that has landed. On the tRNA, you have what's called an anticodon that is complementary to the codon. So in this case, if this codon was CUA, what would be the anticodon here on the tRNA if this was CUA on the mRNA? C with G, U with A, and A with U. That would be the right tRNA. And on the ribosome, there's room for 2 tRNAs to sit side by side. So tRNA 1 lands, tRNA 2 lands, tRNA 2 gets dumped on. tRNA 1 puts his amino acid onto tRNA 2. With the ribosome, ribozymes are going to be the enzyme that catalyzes that peptide bond through synthesis, and it'll hook 2 amino acids together. The ribosome shifts. Along comes tRNA 3. What will tRNA 2 do? Dump his 2 amino acids onto tRNA 3, and then he will leave. Along comes tRNA 4. Then he gets dumped on. Along comes tRNA 5. He gets dumped on, and the amino acid chain gets longer and longer and longer until one of these codons says stop. Oh, we're done then. No tRNA lands. When the shift happens, away you go, and you have a protein chain. That is protein synthesis. DNA determines the mRNA. The mRNA goes out to the ribosome. tRNAs start landing. They don't just land any old place. They can only land where there is a workbench. Where is the workbench? The ribosome moves along the mRNA with tRNA's landing. And whoever is the new tRNA gets dumped on until no more tRNA lands. And as the ribosome shifts, it goes, oh, and we're done. Then a chaperone protein is going to take that chain of amino acids and do what to it? What's the chaperone protein do? It's what you do to your clothes when you pull them out of the dryer. You fold it. You fold it into its right tertiary and possibly quaternary structure. Yeah, here we go. Here we go. Here we go. DNA, start here. What is this? Topoisomerase. Now, remember I told you there was histone and non-histone proteins? The non-histone proteins actually do the unwinding, because you're not unwinding everything. So that's really what I should be telling you. Non-histone proteins, come along here. Okay, now this part's still the same. Unwind and unzip. Helicase. Okay, and what we're going to do is we're going to take our lagging strand, put it away. Okay, this time we're going to go make an R, some RNA polymerase. Just one. You don't have to worry about splice, primer, nothing. Okay, just say RNA polymerase binds to the promoter. Say it.
Binds to the promoter.
That's the next chapter. It's like that's where it docks in. We'll learn that next chapter. Okay, go again. RNA polymerase binds to the promoter. And you go boom, boom, boom. Make your mRNA. Now pull it off. Set it aside. Go get your DNA. Zip it back up. Put it away. Get the mRNA. Okay, here we go. You're going to put on a cap. Put on a cap. And a tail. Here we go. Put on a cap. And a tail. And you cut out the introns. Cut out the introns before it leaves the nucleus. Before it leaves the nucleus. Now it's gone out. Go put it on a workbench. What's the workbench called? Ribosome. What do you call every 3 bases? Codons. And the codons are complementary to the anticodons. And the anticodons are located on the tRNA. What is the tRNA transferring? Amino acids. Okay, amino acids, and the amino acids hook together to make a protein.
Protein synthesis.
Protein synthesis. Okay, got it? I'll show it to you again. All right. Now, look right here. Okay, here's messenger RNA from the DNA. That messenger RNA came from DNA that is coding. That's the coding DNA. It's coding for that mRNA. Here's transfer RNA. Okay, it just transfers amino acids. This RNA has to come, this has to be made too, right? Just like you make mRNA, you have to make tRNA. But this tRNA that's going to go be tRNA and pick up an amino acid, he is from a non-coding sequence of the DNA, and that it's not building a protein, it's just building an RNA. Okay, and here we have ribosomal RNA along with proteins builds the workbench. Also considered RNA that is, this RNA came from non-coding DNA. When you talk about coding DNA, it's coding for an actual protein. Here it's just coding for tRNA and RNA to help in the process of making a protein. So these are your 3 types of RNA. Keep in mind our central dogma, okay? Central dogma says when you use DNA to make more DNA, DNA to DNA, that's just called what? Replication. When would we do replication? Yes, thank you. That's going to happen during the S stage of interphase, right? Which you know all about. Transcription is when you use this DNA to make RNA. And this time you use RNA polymerase. What did we have to use up here? All kinds of stuff, right? DNA polymerase 3, DNA polymerase 1, RNA primase, SSB, right? All those things had to be used. Here we're going to use RNA polymerase and we're going to make some RNA. What do you call every 3 bases on this RNA? And here, now, translation is using this in order to hook a bunch of amino acids together. Oh, how many amino acids do we have? 20. Why do we have 20 amino acids? 20 different R groups. We start hooking amino acids together, and these are called what kind of bonds? peptide bonds, and this happens on the workbench all day. So when you go from an RNA to protein, that is translation. These are 3 key words here. Oh, it's not too much. You're super smart. Damn it. But apparently I can't use a highlighter. Okay. One more time. Yeah. Replication, transcription, translation. Replication, I am making an exact copy. Transcription, think about this. Transcription, I am still in nucleic acid language, right? If I say the words hello, right? I can say that word and then you could do what? Type them up. You're transcribing my words. It's still in that same language, right? Transcribe it. Translate it, hola. Now you're putting into something else, a different language. What's my new language? My protein language. I'm converting it from nucleic acid language to protein language. That's why it's called translation. Not it.
Not it.
Ping pong back and forth. Whoever's going to start first, go. Nucleic acid language and protein language.
Wait, Miss Lynn.
Yes, baby.
So DNA, so RNA.
Wait, let me answer that out loud. I didn't hear you, but let me answer that out loud. Okay, come back to me. Come back to me. Shh, go.
Does RNA go through replication?
RNA does not go through replication unless you're like a RNA virus. So I don't want to just throw it all the way out. But yes. Okay. All right. Big ticket. Do we understand it? You understand all of life now. Do you understand that? Think about it. You know how your code book codes for every protein you see. You know how energy passes through living organisms, how we capture energy from the sun in order to build sugars in plants, and you know how those sugars are used by all cells in order to do cellular respiration, right? So you know how energy flows and information. You control the energy and the information of life right now. You have that in your brain. Go.
Question. All right, so when like RNA is like gonna duplicate, you said that like the lagging goes away. Where does it go? And like
It's just hanging out right there.
Okay.
I just did it because I only have 2 arms.
Okay.
Yeah. Yeah, so it's a part. So RNA polymerase won't dock on to the lagging strand. It'll dock on to the leading strand.
So,
now, controlling what DNA is available for this is huge, and that is chapter 13.
okay.
Yeah.
So I had another question then. Like, if RNA doesn't like duplicate like DNA, then is it just do you just break DNA apart to get RNA? Like, how does that?
Yes,
happen?
that I'm going to teach you that in chapter 13.
All right. I'll get
Remember I said RNA polymerase binds to the promoter?
Yes.
That the promoter is available is huge.
Okay.
Yeah, so I'm going to teach you the control of that in chapter 13. Unless you want to learn chapter 13 and then take your test.
Oh well, that some ideas.
No? Okay. Just throwing it out there. All right. So if we were going to do replication, only, okay? Now replication would happen both on the leading and the lagging strand. But if I was only going to do replication and I'm just looking at the leading strand, what would be my complementary bases here? Do it with me. Now, go. T, go. So you know how to make complementary DNA. So now let's try transcription. What would be the RNA that I would make off of this DNA? Tell me. G-U-A-A-U. What? U-C-C-U-C. What do we call every 3 bases? Codons. How many codons do I have here? 1, 2, 3 codons, right? Now, let's figure out what would be the tRNA that would be the anticodons to this. Tell me the tRNA here. CA what? Right? Look. Okay. Now, this right here, this CAU, it's going to be carrying, this is tRNA, it's going to be carrying an amino acid. Now, to figure out what amino acid that one's carrying, there is the genetic code. They have figured it out. Now, this genetic code is going to use the mRNA. This is the code. What kind of RNA is it? Messenger. You always look at the messenger to find out what the tRNA is carrying, not the mule that's bringing it. Don't look at the mule, okay? People always want to look up the mule. Don't look up the mule. Look up the mRNA. We can look up GUA and we can find out what that amino acid is. Let's go look at that genetic code. Okay? Matat and Nuremberg, they're the ones who figured it out. You used to have to know that, but you don't anymore. So let's go. What did I tell you I'm looking up? GUA, right? GUA. Where are you? GUA is the messenger. So, first base, if I look at this genetic code, I go down to G. So here's G. Hang on, just look this up real quick. Here's the U column. Okay, so all of these are under the U, and here's the third codon, or third letter of the codon, A, right? So now I know GUA. I know what this amino acid is right here. It's valine.
There's a chart.
Right? So you could do that for all of them. Hook amino acids together, you know the protein. Who ultimately decided the series of amino acids here? The DNA. The DNA gave the information to the mRNA, the mule brought it, and we built it. Go review it. Do you get it? Yes. Yay. Super smart. And if you're watching this late at night, have a piece of toast. Cake. Oh, we're having cake now.